I was asked about Wiener filtering of real images. I showed only the synthesized images. So this is from our own research. It's in chapter 10 of the book. We will not go into image deconvolution. But these are SPECT images, single photon emission computed tomography images. And this is of a phantom. I'll start with the phantom image. The phantom is a tube with a solid rod inserted, and the open space is filled with the radionuclide material. Thallium or technetium, it's not mentioned, it's in the book. So the cross section is like a donut or a uddin vade with a hole there. So this is the original image with a lot of noise. Each image is only 64 by 64 pixels. Ideally it should be like that. It's not that, it should be a circle. But this is on a discrete grid. This is after Wiener filtering of this image. This is another filter called the pass spectrum equalization filter. So this Wiener filter is not the same as what you saw. The degradation model is G equal to HF plus eta. There is blurring and there is deblurring here. There is inverse filtering, but with the Wiener filter. Now, this is a 3D image. I'm showing one section. It's a tube lying flat on the table, and then you're taking a section. A tube with a solid rod, and the rest is filled with the radioactive material. Gamma ray photons are emitted. When you take a projection of that, and you get the tube in at different angles, you don't go all the way around. In cardiac imaging, you only go 0 to 180. Now, restoration is possible by taking only this one image in cross-section and restore that. That is called post-reconstruction. After tomographic reconstruction, you have a 2D slice. If you want, you can try in 3D. But we didn't do that. Instead, we said we will do restoration of each two-dimensional projection image separately first. When the scanner goes around, multiple projections are obtained, two-dimensional images of the body, and then the sections are computed. So we can restore each of the 2D images first, and that's better because you take blurring in the other dimension into account. And you can incorporate the dependency of the point spread on the distance between the collimator, the photon counting device. Before that, there is a collimator, and the body going around. So we call that pre-reconstruction restoration. If you restore each of the projection images first, and then do the tomographic reconstruction and get the cross-section, and then, CT, you get this. Compare that with this. This is Wiener filter applied to each projection image, and then reconstruction, and a few other options. So these two are Wiener filters to this image, and this is to each projection image. One of my PhD students did that, and this is of a patient. This is imaging of the left ventricle. This is one of the projection images at some angle. So for this type of imaging, they go 180 degrees here, not the other side. From here to the other side, 180 degrees. And that's one of the projection images. The heart is here. The abdominal organs also have taken up the radioactive material. There's also the intravenous tube there. Well, you're interested in this part, and that is reconstructed in some 3D, and some other planes are cut. So these are the images corresponding to that phantom I showed. If there is a blocked part, then that part of the left ventricle is not getting blood supply due to some blockage. So here we have I need to check which is which. Post-reconstruction using the Wiener is B. A is the short axis SPECT. This is the original. This is after Wiener filter. Pass spectrum equalization METS filters. But if you apply the Wiener filter to each projection image, which is that 2D image you saw and then do reconstruction, we get this. Certainly this is cleaner than that and sharper. So this is a real life patient's image using the Wiener filter. And we have done the Kalman filter. In these cases, the images are fairly small, spatially. When you consider the liver and the abdomen, then the object is larger, and you need to take into account spatial variation of the point spread function. And then we have used the Kalman filter. And I won't go into that because we are not looking into the Kalman filter. But just for the Wiener filter, I wanted to show that. We tested that with the Phantom that we built ourselves in our lab, and then with real patient images. This is another Phantom. This is another Phantom made by a company where there is a drum and a number of tubes here. If it is white, it means it is hot with the radioactive material in the tube. If it is not active, it means there is no radioactive material in the rest of the drum. And there are multiple pairs of these tubes. You don't see these at all. They're all blurred out and lost in the noise. There are two big tubes here, and there are multiple tubes here in pairs. After restoration, you start seeing some. And then some more. 1, 2, 3, 4, 5, 6 pairs of tubes. And it's cleaner. D, which is which? B is Wiener filter. This is pass spectrum equalization. This is the Kalman filter. Just so that you see application to real life images. This is the liver and spleen. Before, this is the original, and this is using the Kalman filter. Sharper edges. Are these real? We don't know. The radiologist looked at these images and said there is some enhancement, but we can't quantify it. They did not do any diagnosis and diagnostic evaluation with multiple patients. We didn't have the resources to do that. Another case, the original, and this is with the Kalman filter. But here, the model is G equal to HF plus eta, blurring and noise. So there is deblurring and noise control. If you want to read more about that, you go to chapter 10 in the book. I don't have lecture notes for this to give you. Okay, let's go back to our lecture material. We saw the Wiener filter and the adaptive LL-MMSE filter. Now there are a few more adaptive filters where some parameters are modified as you go pixel to pixel. The adaptive 2D LMS filter, least mean squares filter, is like a fixed window Wiener filter. The window size is fixed, but the filter coefficients vary. You apply this in the space domain. But as you go pixel to pixel, the coefficients within that mask of the filter will change, will adapt to what is present in the image. The adaptation is based on the method of steepest descent, and it tracks variations in the local statistics of the given image or signal. In my signal processing book, you will see the same filter for biomedical signals. It does not require any a priori information about the image, statistics, and so on, correlation and all that. There's no averaging, differentiation. It just keeps on adapting. There are limitations. It's a nice approach, but it doesn't always work very well. So assume we have a causal FIR filter, finite impulse response filter, WLPQ. L is an index related to the position in the image. Mn are the pixel locations, PQ are the locations of the filter coefficients. This is the convolution integral on WLPQ. G is the given image, n minus p, n minus q. And at output f tilde mn. So L is the current position of the image as you move from along one row, column to column, and the next row, and so on. L is given one index, m times capital M plus n. M is it's an m by n image. So as you go one row to the next, this is a running index, but one value, not two. So L goes 0 to mn minus 1 for an m by n image. So the filter coefficients for each pixel location are determined by taking the filter coefficients at the previous location and modifying them. And what is the modification? You take WL plus 1, the new filter for the current pixel location, is given by WLPQ, the filter for the previous location on the image, minus mu, this gradient operator nabla E square, L, where mu is a scalar that controls the rate of convergence and stability. You don't want the filter coefficients to vary rapidly. If you make the constraint too tight, then the filter will not be able to adapt. If the filter changes rapidly, there could be instability. So convergence and stability need to be controlled, and that's this factor mu. EL is the local error signal, and nabla is the gradient operator with respect to WLPQ. We will see how to get that. So E is defined as D minus F tilde, where D is an approximation to the original FMN. Dr. Narasimhadhan was asking that question. In 1D, this is how it's been derived, even for the Wiener filter. That you assume that the original image is known, and then you say, well, I'll make an approximation, and I'll take it to be a delayed version of the current distorted image that you have on hand. So DMN is an approximation to the original. And what is DMN? You take the current GMN, the distorted image you have, with a delay of one samples, one sample before and previous row and previous column. Why? I don't know if that's a good approximation. That's one of the assumptions made, but that could be noisy too. How can that one sample before be a good estimate of the current pixel without noise and without distortion? But that's the assumption made. So WL plus 1 PQ is the current filter, the previous filter, plus 2 mu, this error, and GM minus P, N minus Q. So that's the model. It's a recursive equation defining the filter coefficients of the pixel location L plus 1, the new location, with reference to the previous one. So here is that shapes test image. It's a different one. We have multiple versions of this. This is Gaussian noise added. This is the 2D LMS filter. And this is the next filter that we will see. So look at just this one now. The other filters will come later. So from there to there, there is limited reduction of noise. The edges are retained quite well, but there is still a little bit of an overall blurry appearance. So I wouldn't rank this filter very high. RMS 14, actually it increased from 14 to 15. The error with reference to the original. So the output of this depends upon these parameters. How do you control these? So it's a bit difficult to optimize that any further. There is another adaptive filter called the adaptive rectangular window, where the window itself is adapted. How big do you want that window to be? To compute the local statistics, we said 2p plus 1, 2q plus 1, rectangle window. what should be the size of that? Here they said adapt it. Let it vary in a uniform area where there is no noise and no structural information. Make the window as big as possible, use as many pixels as possible, and get a good estimate and suppress noise. As the window comes here, there is some edge, some information about the signal, then you reduce the window size. So as you move here, window size is reduced and adapted so that you don't mix. You don't take a big statistical average over a large area. So this was proposed by Song and Perlman, and then there are other references from their group. It is assumed that the image processes have zero mean, which is not true, so you subtract the mean. Given an image, take the mean and subtract it, store it because you want to add it later. Otherwise you will have negative values and edges only, and you don't want that. So F tilde mn is some weight alpha mn times gmn. Previously we had A, G plus B. The plus B is removed now. So you want to find alpha mn at each pixel using some criterion, minimum mean squared error, for example, as in the Wiener filter, or something else, but they use the same. EMN is F minus F tilde, F minus alpha G, similar to what we have seen before. So minimization of mean squared error requires E to be orthogonal to the image. So expectation of F minus that times G should be equal to 0. That's the requirement imposed here. So then solving for alpha, you get this. You have seen a similar ratio before in the Wiener filter. Now it is variance of the original image, variance of the original image plus variance of the noise. So this is the variance of the given observed image. Because a noise is of zero mean, the a posteriori mean, mu g, is equal to the a priori mean, mu f. So the mean of the given noisy image is equal to mean of the original. So then F tilde mn is mu g, not mu f, but now you say mu g. And this factor times g minus mu g. So this is similar to what we had seen earlier. A low pass filter, a high pass filter, and a weighting factor. Okay? Some small variations, but the basic idea is the same. How do you implement it? You assume that pixels in the immediate neighborhood of Mn are samples from the same ensemble. So there is stationarity. Global non-stationarity is now considered to be locally stationary. The whole image is non-stationary, but in small portions it is stationary. That's the assumption in that window. And ergodic. So you replace ensemble statistics with spatial statistics. So if we determine the size of a neighborhood in which the image values have the same statistical properties, the sample statistics can approximate the posteriori parameters in that equation we saw. So you need to identify the size of a stationary rectangular region within which to compute the statistics for that pixel. The window size changes according to a measure of signal activity. So they define some signal activity. Signal means image structure. So the adaptive rectangular windows were defined to be of odd length LR in row and LC in the column direction. So LR is 2 NR plus 1 and LC is 2 NC plus 1 to represent they are odd. So you go minus NR to plus NR, minus NC to plus NC to take the local mean, just adding the pixel values in the window. And local variance is the pixel value minus minus the mean and the summing and averaging. Right? You have seen these before many times. Now, these are varying pixel to pixel. This is varying pixel to pixel as you filter. Sigma f, sigma square f, the variance of the original image is the variance of the given distorted image minus variance of noise. We said earlier that Sigma square G is sigma square F plus sigma square eta. Now it's the other way. So you are estimating the variance of the original signal, original image, by subtracting the variance of the noise from the given noisy image. So you need to know noise variance. As I said, noise variance you estimate somewhere else from some part of the image that has only noise. Then the estimate is mu G plus G minus mu G. The same thing, low pass, high pass, with this weight factor. Sigma square F is given from that. Sigma square eta must be independently estimated using some other mechanism. These are computed for every pixel. LR, LC, NR, NC, mu G, sigma square G, sigma square F. To vary the size of the window, they recommended a signal activity SRMN in the row direction. R is row, where they took a kind of a variance in the row direction. This is variance. This is mean in the row direction only. Minus NR to plus NR, M plus P only, N remains the same. Minus sigma square eta. So this is, you are comparing the signal variance, subtract noise variance, and the mean is local in that row direction only. And they said NR is reduced if the signal activity is more than some threshold, or it is increased if the signal activity is less than the threshold. And the threshold was defined as some factor kappa times sigma square eta, noise variance, divided by LR, the length of that window. So there is some factor kappa to control. The same is done for the column direction, similar. So the rectangular window adapts by becoming broader or taller as you go pixel to pixel, and the local statistics are computed for every pixel as the window moves. I'll go through this filter and then we will go back and look at the results. The adaptive neighborhood filter is our own design. When I went to Canada as a postdoc fellow long ago, 44 years ago, 1981, I went as a postdoc fellow. My supervisor, Dr. Richard Gordon, came up with this idea that we should have adaptive filters that don't depend simply on fixed windows or populations of pixels as you move the filter from pixel to pixel, but instead the neighborhood of pixels that you use at every location must adapt to what is present in that location. So we call that an adaptive neighborhood. Not rectangle, it could be of any shape and any size, depending on what is there. So if you're processing a pixel here, it should take this, say, approximately the shape of that. If you're processing a pixel here, it will take the pixels there. You don't mix an object with the background. That's the whole idea. But there's not always a background and an object clearly. In a mammogram, there are so many parts. But there are some regions that you can identify, like calcifications and ligaments and ducts and so on. And there are some background tissues, fatty tissues and others. And you don't want to cross their boundaries. Don't blur across boundaries. He wanted to call it pixel-independent processing. That is, we are not processing pixels, we are processing regions or objects in the image. But there were other papers, other methods called region-based or, what is the other one I said? Object-based processing. So we could not use those names. So we said pixel-independent, and even that was criticized, so we simply said adaptive neighborhood. So a variable size, variable shape neighborhood is defined or determined rather for every pixel in the image. It's expensive computationally. The adaptive neighborhood grown for the pixel being processed, which we call the seed, contains only those spatially connected pixels that are similar to the seed. There's some similarity like a distance measure. The neighborhood does not grow over edges but overlaps a stationary area. The statistics computed using the pixels inside the adaptive region are likely to be close to the true statistics of the local signal and noise components at that location. So when I'm processing here, I can grow a region that is like this, however big I want, taking all the background. It could be of some shape and size. But I can put limits. I don't want 10,000 pixels. I'm happy with maybe 100 pixels. That's enough to compute statistics. So even though I can grow a huge region for a pixel here, I may stop there. Whereas for this, I'll grow a region that takes only pixels within that object. So region growing. For every pixel, you have to grow a region and find what is the local region, what is the local object. So you compare the 8 connected neighbors GPQ and the seed GM. GMN is the filter, the point where you are filtering. GPQ are the neighboring pixels, but not just left and right and top and bottom, but somehow connected. The connectivity is determined by, the similarity is determined by this DPQ, just the difference. And we have some modifications to that I'll show later. So pixels GPQ having DPQ less than some threshold and spatial connectivity will be included in the region. The procedure continues by checking the neighbors of this new pixel you added in the neighborhood and take their neighborhood and ask, can I include them and their neighbors? And all connected neighbors are checked. Until you hit some boundary, you cannot grow any further. So adaptive neighborhood is grown for each pixel in the image. That makes it expensive. Now, once you have this region, in some types of filtering, you may want a reference around that. So you put a ribbon around it of the same shape and take those pixels to represent the background around that region. There is the notion of redundant seed pixels. When I have a region grown from here, many pixels here may give you the same region again and again. So we say once you grow a region, if you use a simple criterion for inclusion, then you can say that if I were to take another pixel from that same region, I'll get the same region as the adaptive region. So all of those pixels will get the same output. I don't have to repeat the process. So we call them redundant seed pixels because you would get the same output if you do the entire process. It depends upon the threshold used for region growing and all the conditions you put. It cannot be done always. So here's an example that my student William Morrow developed, a nice illustration coming from Canada. That's a mountain in the Canadian Rockies. I don't remember its name. There's some snow on the mountain there. And that's the pixel from where region growing is starting. And these are the gray pixels are the region pixels being included in the adaptive neighborhood. The black pixels have the same value as the starting pixel, so they are the redundant pixels. And if you grow again using those pixels, you get the same output, same result, same region. So you don't have to repeat. So the region is growing, and here it has hit the boundary. It's not growing anymore in that direction, but it grows here. And that's the final region. And around that, in dark gray, there is the ribbon, which will be the background for that region. So the starting pixel was somewhere there. All these black pixels have the same value as the starting pixel and are redundant seed pixels. These gray pixels are included in the region, and there could be some holes here that don't belong. Some pixels may not belong, but they are inside. We don't use them in computing the statistics. And that is the ribbon all the way around. You can define a certain thickness and obtain it by dilation of the boundary. You do that for every pixel. So for a pixel somewhere here, you may get a region like that. For a pixel here, you may get a region like that. So then what do? You compute statistics. Why 3 by 3? But the basic question we asked is, What is so special about 3 by 3, 5 by 5? They're not sacred. And similarly, why rectangle? Why not circle? Why not something else?
Easy grid is 3 by 3.
What's that?
Easy grid is 3 by 3.
It's easy, yes. I said it's not sacred, but 3 is sacred. Yeah, there are many things 3. So that's the basic question we asked. Why square and why fixed? 3 or 5 or 7 or something. And why odd? Odd is for centering. Here it is not centered. It could be any shape and it could go somewhere else. But because of the constraints of spatial connectivity and similar gray level that we impose in region growing, the assumption or the model is that all of these pixels belong to some object with similar properties. So when you compute their statistics of those pixels, then you are getting the statistics of some signal component, image component that is present at that location. But that object has some spatial extent, so that could apply to other pixels in the same object. But the objects are not constant. So for another pixel in the same object, you may get a different region. It's not segmentation. But for every pixel, you grow a region. But under certain conditions, a number of pixels with a given region may get the same result. So here is one of our shapes image, and with Gaussian noise added, and 3 by 3 mean, 3 by 3 median, adaptive neighborhood mean, adaptive neighborhood median. There's a little bit of blurring. Now we compared with 3 by 3. It's unfair. We have compared with others later. So the original and noisy, 3 by 3 mean and median. Obviously the 3 by 3 mean is blurring. 3 by 3 median may have caused some distortion. It's a little hard to see here. And the adaptive neighborhood mean and median. There are still some regions where, some parts where the region growing has not done well. But there is no blurring of edges, even with the mean. And one of my postdoc fellows, Raman Parangate, he came up with the idea of noise subtraction. Instead of estimating the signal, he said, we will estimate the noise. It just reverses the process, that's all. So this is similar to the adaptive rectangular window LMS formulation. So there is not much to do here except that eta is estimated, signal is estimated eta tilde as alpha GMN. But then it leads to something very similar as before. F tilde is G minus eta tilde, and F tilde is beta GMN, but beta is 1 minus alpha. So the rest is the same. So I'll skip all of that. In the end, you get a result that is very similar to what we have seen before, but 1 minus that. So let's skip that part. There's a flow chart, but I think I have already explained that. So here is another image. Is it blurry? To me, it looks a little blurry. You said okay, but it is a little blurry. So here is a part of the peppers image. This is part of a pepper, the edge, noise added. That's the seed pixel. This is the region grown. With some adjustment, this white is the adaptive region. It has not crossed the boundary. And these pixels are not included. There were some pixels inside the region that later on were included. Another postdoc, not postdoc, but he was a visiting researcher, Mihai Chuk, from Romania. He spent some time with me and modified the region growing procedure and put some constraints so that you don't get huge regions. We don't need such a big region. So if we get about 100 pixels or so, it's enough. So we stop region growing there. But on this side, it stops due to the conditions implied. Conditions imposed, rather. So after filtering, we get this from there to there. This is only a part of the image. The whole image was filtered. So this is the noisy image. This is the filtered image. So this is with the test image, the clock image. So 3 by 3 mean, median, the refined LLMMSE filter, and sorry, not that. D is the refined LLMSE filter, and there was another noise updating recursive Wiener filter that we didn't talk about yet. This is the adaptive neighborhood filter. So this is the noisy original image. We didn't add noise. That's the way it is. That is 3 by 3 mean, 3 by 3 median. We had seen that earlier. So we have good noise reduction compared to that, and all of these numerals are preserved better compared to the 3 by 3 filters. The others are not bad. So Mihai did many of these experiments comparing various basic filters and advanced filters, all the adaptive filters. I don't have to go through all of them. You could go through them later. The Peppers image, this is another commonly used test image. The noise level is too low. Let's skip that and this is Poisson noise, where you can see a little better the noise that is present. And C is refined LLMMSE, the noise updated recursive Wiener filter. E is adaptive neighborhood. This is our mean filter, and this is our adaptive neighborhood LLMMSE filter using adaptive neighborhoods. We showed that our methods gave lower RMS errors, cleaner images for various types of noise with not much of blurring of the edges.
Question here is like, just my personal feeling also. When we see MSC 85.32, when we do research and if we get some result, what happens is 85.3 and 86 point something. We don't know whether our result is very much improved or not.
Correct.
If we get a little bit increase in the number, we tend to publish that like.
Yeah. The question is, how many decimal places will the result be relevant? Is 85 better than 86? That's like 1 point some percent. It's not a huge difference. But perceptual evaluation matters more, and it's been shown that mean squared error and RMS don't agree well with human perception. There are other measures. So look at this and see if you can focus. So let's wait for it to be focused. This is the salt and pepper noise illustration. So we showed this, and Mihai did a detailed study, and we have all these measures. I don't want to go through this table comparing error measures. We will skip these and maybe just look at one of these pictures. You tell me from there. By the way, that itself is a good filter, a low pass filter when you blur the lens. Really? Yeah. Oh, you see there you have a zoom there. That's good. Let me put up text here, there. Let's look at the text. Oh, blur. Okay.
This is a maximum.
Okay.
Okay. Better.
Better? Is that okay? But if you look at this image, some of them are blurry, so it's hard to judge. And it's okay. You may take these files and zoom and take a look in detail yourself later. So this is with salt and pepper noise, and C and D are 5x5 mean, 3x3 median, and adaptive neighborhood mean and adaptive neighborhood median. So we have shown it works reasonably well with multiple types of images and also with color images in a different paper. And there is a table of mean squared error values. I won't go through that table. It's not necessary. There are a few other examples where multiple methods have been compared. The clock image, you have B is 3x3 mean, 3x3 median, refined LLMSE, the noise updated repeated Wiener filtering, and the adaptive neighborhood LLMSE filter. So in this way we compared and did an analysis. And evaluated multiple filters. But eventually, the user in the application domain needs to assess them. We use the error measures, but error measures are not correlated with visual perception. These measures. There are other measures in color images using other domains. The LUV and LAB domains have been shown to be better correlated with human perception, and error measures defined in those domains have been shown to be better correlated with human perception. But in grayscale images, we don't have any other representation. We just take the LMS and mean squared error. I mentioned earlier that there are the Laplacian errors and then the visually perception, human visual perception related filtered error measures. So you could use those and compare, but we haven't done that. It all depends upon which model suits the application. Okay, at the end of each chapter I have given some applications. So there is a bit of a repetition in the sense I showed some examples earlier with synthetic images. Now I show the application. Multi-frame averaging, I mentioned earlier, ensemble averaging could be done to reduce noise. In confocal microscopy, you use a laser beam to scan a sample. It could be a biological specimen. There could be some thickness to it. So a laser beam is scanned, and image, it finally focuses on certain planes, and the sample is tagged with the fluorescent dye, and that gives out light at a certain wavelength. The use of coherent light obviates the blur caused with full white light, the full spectrum. So you use only that specific wavelength of light because the laser beam is having just that frequency. Laser excitation causes the dyes that have been applied to the sample to emit light, that is fluorescence. So the sample fluoresces at a particular wavelength. And using multiple dyes to stain different types of tissues and structures in the same sample will lead to a color image with different parts emitting different color when triggered or excited by the input laser beam. So the confocal microscope uses a pinhole and then focuses on one plane. The sample could be thick, a few millimeters, not that thick, but a few millimeters, and a certain plane is focused, and then by changing the focal plane you can get multiple slices. You don't do 3D really, but you could stack up those slices. The light from other planes are blocked. Light from various other planes, only one plane is focused. A photomultiplier tube is used to amplify the light, and that could cause some noise amplification. So you average, you take the image over a certain number of time and certain window of time and take a number of images. A scanning mechanism is used to raster scan because it's a single laser beam. You have to scan the sample and then assemble the image. The sample step could be of the order of 0.1 micrometer. That's the resolution you can get. The distance also matters. The planes, between the planes, the separation could be of the order of 1 micrometer. And it is possible to assemble them to get 3D, but I didn't see any 3D examples. What I got is just one plane at a time. The sample I have here is a set of cells from the nucleus pulposus, which is the portion between the intervertebral discs, or the discs between the vertebra and the central portion of the discs, scanned using a laser beam with that wavelength of 488 nanometers. The red dye that is used gives radiation or light in the frequency of our wavelength of 585 nanometers, and that corresponds to the nuclei of the cells. The green dye in the band of 505 to 530 nanometers shows the filament structure. The focal plane thickness is about 1 micrometer. Now, if you take just one image, it'll be noisy. If you take multiple frames, the sample is fixed. It's a biopsy specimen, so it's going to be there. But if you scan it too many times, you're depositing laser energy into that. There could be local heating, and the sample could change its characteristics. If you keep scanning, it could lose its capability to fluoresce, or it could develop autofluorescence. So there are other complications. So even with a fixed specimen, you cannot do this a million times. So you can acquire a few images and average. So here we have the red dye and the green dye showing the filaments. Here you see the cell structure, cell nuclei, the actin filament structure, and when you combine them, you get the color image. It's an artificial color image due to the excitation with the laser. This is the light emitted from the sample by fluorescence. The width of the sample is 145 micrometers. Let me see, was this averaged? Each image was averaged over 8 frames. So here we have just without the color, and let me read the single frame acquisition and another single frame, averaging of 4 frames and 8 frames. One frame, another frame, average of 4, average of 8. So if you compare 1 with average of 8, you see some reduction of noise. The details are not any a little sharper, but the noise is reduced. So these are real acquisitions of a real sample. The noise is inherent in the process of acquiring the image, and also from the photomultiplier tube. When you amplify the signal, noise is amplified. Okay? Now, when you filter a single frame acquisition image, suppose you cannot acquire multiple frames, you could try filtering one frame, 3x3 mean, median, 5x5 mean, median. You see blurring. The noise is reduced. The median is better, but the mean blurs the edges. Again, there could be some blurring in the projection. If you take the original PDF file and zoom in, you can see the details. So we did many of these. We applied many of these filters and tried the LLMMSA filter, refined the updated recursive Wiener, adaptive neighborhood. This is our filter, and it's hard to compare, but you could compare, and we cannot compute any RMS error here because we don't have an original. We did some Fourier domain filtering. This is just to try all the filters we have studied so far, most of them. And you could look through them and come up with your own opinion as to which one does a good job and which one doesn't. So I'll stop the comparison here. In nuclear medicine imaging, I mentioned earlier about gated blood pool imaging. This is also a kind of multi-frame averaging. Depends on how you view it. So nuclear medicine images are typically acquired under low photon conditions because of the radioactive material that you inject into the patient. You have to be concerned. You have to take into account the bad effects of the radiation. For a number of hours after that, the patient will be still emitting gamma ray photons. It'll take some time for that to be passed out, to be excreted. Counting of photons emitted over long periods of time reduces the effect of noise and improves the quality, but then imaging over long periods may not be feasible due to motion artifacts. And also the patient won't sit there for 2 hours. So here is an example with that phantom I mentioned. The geometry of the phantom is shown here. This is just an image. So there is this drum, plexiglass, and 2 tubes that are filled. Actually, it's a solid cylinder with these holes drilled. So in the holes, you put the radioactive material. So there are 2 big holes and pairs of holes like this. 1, 2, 3, 4, 5, 6, 7, 8 of them actually. And these are, the dimensions are there in the book. I won't go through that. So if you acquire these, in this case, we can count the photons for a longer time. So the first one is 2 seconds, 15 seconds, 40 seconds of counting the photons. So the longer you count, the better the quality. In 2 seconds, there is nothing really. It's so noisy. Even here, you cannot see the very small ones. There's a lot of blurring. The resolution is poor. But compared to that, it's clean. Compared to that, it's clean. And this is gated blood pool imaging, where the ECG is taken. The patient is administered this radioactive material. There are 6 phases of the cardiac cycle here. The heart, the left ventricle. The left ventricle is contracting, the smallest here, and then it is expanding back. The size of the left ventricle and how much blood it is pumping out is of interest. How much blood is remaining after full compression and how much it is with relation to when it is filled. A certain amount should be ejected out at every compression. If it's not ejecting much of the blood, then there is a problem in compression. Now, how is this obtained? There's still some blur. The ECG is taken. The QRS complex corresponds to contraction of the ventricles. So this is systole, the beginning of systole. So you detect this using techniques, as in my other book. I'm promoting my other book, which is not here, but it's in the library. Paperback editions of that book have been published here. I don't know why they didn't do the 3rd edition yet. The 1st and 2nd editions were published by Wiley, India, in paperback. But anyway, detection of the QRS complex is not a big thing now. That was part of my PhD thesis project anyway. From 1976 to 1980, that was current research then. Now your smartwatch or something does it. It has become a consumer electronics thing. So you detect the QRS and then put a window there and say for those, how many ever milliseconds, when the photons are emitted, I'll put them in this frame. That's an image. So all those photons It's a 2D camera, the gamma camera or anger camera. So it acquires the image and puts it there, and the next window of how many ever milliseconds, you can divide the cardiac cycle into 8 parts or 6 parts, not 50 parts, and then take that and put it here and the next and here and so on. And when the next beat comes, that QRS complex, that window data will go to the same phase as for that beat. So for every beat, when the QRS comes, the data will come to the same buffer. So if you take one such, you can say it is one frame. And when the next beat comes, you can call it another frame. So then you can call it multi-frame averaging. You don't have to divide. If you want, you can divide, it doesn't matter. It is accumulation of the counts. Is it multi-frame? A sort of multi-frame averaging. And these were averaged over 16 cardiac cycles. Each one of them was averaged over 16 cycles. I couldn't get one cycle data. They don't save it. So I cannot show you what it would be if you acquire only one cardiac beat. It was not available. But anyway, with averaging over 16 cardiac cycles, you can see the ventricle changing its size and shape. There is some accumulation in the thyroid and other parts. The rest of the body also takes up some activity, but not as much. It is tagged to go preferentially into the heart. Okay? You said 3 o'clock, right? Okay. Okay. Any questions about removal of artifacts? We have seen a number of filters.
I was observing like this. For example, what we process filtering everything for computer processing. It's not for, there is that helps the doctor to see and diagnose or whatever it is. But finally our computer processing is for computer understanding.
Okay.
Because computer can understand some pixel value, but human may not perceive that.
Yes, we don't consider pixel values. You don't sit and read each value. You look at the whole picture, and our visual system is good at doing that. These images I'm showing, I'm not showing any medical application yet of noise removal. What I've seen is noise is not a big issue in many medical images, including mammograms in particular. They said, okay, it looks nice and clean. I don't need any filtering. Noise removal always comes at the cost of some smoothing, and they don't like smoothing. They don't like edge blurring, especially. Radiologists are even happy with these images. These noisy 64 by 64 pixel images, they do diagnose with this. If you filter that and there is blurring, they don't like it. They're used to that. It's a question of, the point is they're trained with that. Okay? They're trained with that and they can detect it and they can do that very well. And if I go and say, I filter it, I look at this Wiener filter and all the fantastic things, they say, yeah, the noise is reduced, but you blurred the edge. And I don't want that. So even a little bit of blurring of the edge, they do not tolerate. So noise removal in all of our work has not been found to be suitable for medical image interpretation by the radiologist. It's a pre-processing step, both for computer analysis and for other steps after which you may give the image to the radiologist. So the next chapter is on image enhancement, and I'll show you assessment of mammograms with enhancement. We did detailed studies of enhancement, not noise removal. We concentrated on mammograms because I got research grants for that, and I had a collaborator, and we collaborated with the breast cancer screening program in Alberta, and nobody was interested in noise removal. They were interested in contrast enhancement. And we with many radiologists, we did detailed analysis and we gave our program to others and they did analysis of enhancement, contrast enhancement. Contrast is always the issue. But when you do contrast enhancement, you have to take care of noise. You don't want to enhance noise. You don't want to blur edges. So those are required. So controlling noise becomes part of contrast enhancement. This is also enhancement. But the focus is on removing noise, not sharpening. Next, we will see how to sharpen edges if that's what you want, or to enhance contrast if that's what you want, while controlling noise and while controlling unwanted blurring. But typically there won't be blurring there, there'll be enhancement of noise. When you enhance contrast or sharpen edges, noise also gets boosted. That's the price to pay there. Here, you want to suppress noise, you lose sharpness. Opposite. There have been other studies. I'm not saying it's not important. There have been other studies in the literature on removing noise in mammograms too and evaluating them. But you're right that some of these steps are for subsequent processing of the result using computers. Some of them are for subsequent processing by human experts. So depending upon what the next step is, you have to choose an appropriate method that suits that step. Okay? So I call this next chapter image enhancement, but to be specific, you could say edge and contrast enhancement, or improving the appearance of the image or appearance of some aspects of the image that are of interest. So in many practical applications, there are some issues that will limit your capability of getting a good picture. You cannot blast the patient with more and more X-ray photons or inject more of the radionuclides or take the picture over a longer period of time or make the patient stay in the MR scanner for longer. There are limitations there, and you always sacrifice. Patient safety and well-being comes first. If you get a poorer image, then you have to deal with that. So what happens? You get less than the desired levels of contrast, visibility, of detail, or quality. What is quality? We said earlier, we need to characterize quality in terms of high frequency content, acutance, sharpness, contrast, and so on. We saw a few measures. So in several situations, the understanding of the exact cause of the loss of quality is limited or non-existent. You may not know what is causing this blurring or loss of quality. An attempt to improve the quality of the image using several techniques applied in an ad hoc manner, meaning I'll try this, I'll try that, if one works, fine. So then that is what is called image enhancement. You're not applying any optimization criterion here. It is not restoration. When you apply a specific model of degradation and have a specific optimization technique, like the Wiener filter for deblurring I showed, then that is called restoration, image restoration, with deblurring and denoising. When you don't have such criteria and you simply say, oh, I like 3x3 mean, median, this, that, try all of them. If any one of them or two of them work, then be happy. That is the ad hoc manner of image enhancement. Some of them do have models. It's not like they are totally, baseless. They have a basis. Some of them have a basis, but does that suit your application? That we don't know in some cases. So in some applications, I say a non-specific improvement of the general appearance of the image may suffice. Make the image sharper, let the details come up a little better, and the user is happy, fine. What are these situations? Microcalcifications in mammograms, lung nodules in chest X-ray images, the vascular structure of the brain. If there are very fine fractures in the ribs, it's hard to see them. If you can enhance them and make them visible, assist in the diagnosis and detection of them, then something is achieved. So some could be difficult to see due to small size, subtlety, small differences with respect to surrounding structures, low contrast, superposition by other tissues, as I said, breast tissues, normal tissues, and calcifications and tumors. They're all superimposed, then the calcifications may not be visible. So what is desired is enhancement of contrast, edges, and detail visibility without causing any distortion or artifacts. So you don't want to cause something else to appear, leading to false diagnosis. So a basic enhancement technique is digital subtraction angiography. It's just A minus B. So just subtraction helps. But to achieve that, it's not easy. I was telling this to one of my PhD students I had worked with a medical imaging company. I said, well, why is a digital subtraction angiography system so expensive? You're taking one image, you're taking another image later, and you're subtracting. It's not like a CT machine where we have all these algorithms. He said, no, there are some complications involved, and it is not so simple. Okay. In DSA, or digital subtraction angiography, you take a picture of the patient first, and then you inject a X-ray contrast medium, an iodine compound, which is heavy substance, so more attenuation, and it fills the blood vessels, and the blood vessels will attenuate the X-rays more than before. And the change is only in the blood vessels. So you take a picture after, a little after the injection has been done, and the contrast agent has settled in the parts of the body that you are interested in. Take a picture and then subtract the two. So the difference is only in that injected contrast medium, so the blood vessels will show up. A number of images are taken. I don't know how many they take. A few, I shouldn't say a number, a few images are taken as the agent spreads through the arterial network and before it is dispersed and excreted out, so that will take more time. So within a few seconds, you do this. An image taken before the injection is used as a mask or reference, and you subtract that out from the live images where the contrast agent is coming through. So, being an engineer, I wanted to put an equation for everything. I don't give lectures without equations. And I was told, this is a plenary lecture, just talk. I said, no, I want to have at least one equation. So A minus B, F1 minus F2, if you want to write an equation. But there are artifacts due to movement. Even within the few seconds, the head you can put a clamp and hold it. But other parts of the body, you'll be moving a little, and that causes some artifacts. And even within the head, even if you hold the head steady, the arteries are pulsating due to pulsating flow of blood, and that causes artifacts. You cannot hold that. And allergic reactions to the contrast agent. Some people develop allergic reactions, some may even die. There have been cases in Alberta. There are different types of these contrast agents, and the government, because healthcare in Alberta, in Canada, is funded by the government, funded by me, taxpayers. They are spending our money. But it is controlled by the government, by the health authority. They determine how much to spend on this and that. And there are different types of contrast agents available. Some are, some cause allergic reactions. They are known to cause allergic reactions to a larger population, and some have a lower level of that risk. And the government has to balance between the two. The healthcare authority has to. And there was a dispute as to which one they should use. So that is a risk. There is a cost-benefit analysis everywhere. So this is from one of my friends in the Netherlands. They gave this image. These are the two images before and after. So this is The brain, you can see the midline, the socket of the eye. You cannot see the vessels here. After injection, you can see the vessels here. So the difference is very small. It's not like it'll just pop up like the bone here. That, and see here, you don't see the blood vessels. You see a few here. You see some here, but those, I can't see them here. This is the subtraction, but there are many of these artifacts due to motion. And even the blood vessels, there is some motion here. They have a motion correction method, and this is after that, which is clearer. You don't see these artifacts here. This is from the Netherlands by Meyring and Zuiderveld and Veergever. There's an interesting dual energy and energy subtraction X-ray imaging technique that was developed in Chicago. The subtraction technique I showed just now, you have to take multiple images, one after another, at least two. That means more radiation exposure. In this technique, there's only one exposure, but they put filters in the cassette with the film or detector, and there are multiple films or multiple detectors with copper or aluminum filters in between. The filters will stop some of the soft X-rays. The X-rays have already passed through the body, but within the spectrum of the X-rays, there are low energy photons and high energy photons. High energy photons have already come through the body, and some low energy photons would have been absorbed right away at the skin. Those are not important. Others have been attenuated by passing through the soft tissues. They contain important information. But you want to separate that spectrum. And how they do that is by putting a copper plate in between the film. Suppose they're using film, 2 layers of film, 1 filter. And the filter will stop the soft part of that radiation that has already gone to the patient. The hard X-rays will go through. The hard X-rays would be related to denser parts like bone, soft due to soft tissues. So they get multiple images like this, at least 2, and then take a weighted combination of them. Depending upon the weights you apply, you can emphasize the soft tissue or the hard tissue. So X-ray measurements, now remember that different materials have varying energy-dependent X-ray attenuation coefficients. So the X-ray attenuation coefficient of this piece of tissue depends upon the energy of the beam, the photon energy. And the X-ray beam we get from the X-ray tube has a fairly wide spectrum. There is a filter there, but still it has a wide spectrum. X-ray measurements or images obtained at multiple energy levels, known as energy selective imaging, could be combined to derive information about specific materials, not elements. In some cases you could, but here soft tissue and hard tissue. There are some techniques where you can get information about the presence of certain elements. Weighted combinations of multiple energy images may be obtained, in this case, to separate soft tissue and hard tissue details. Typically, to do this, you get multiple images, but that has the disadvantage that the patient has to be exposed repeatedly at different energy levels, and that is the kilovoltage in the tube that is applied for the acceleration of the electrons. And then there will be motion artifacts. So in this technique, there are multiple plates separated by a copper filter. So they used plates that give you a distribution like a charge distribution. It's indirect digital radiography, that's what they used. The first plate gives you the full spectrum of what has come through the patient, plate or film. The copper filter passes only high energy photons of what is there, and the second plate takes a picture of that. Bones and calcium-containing structures would have preferentially absorbed the low-energy components of the X-rays. And high-energy components would have passed through low-density tissues without much attenuation. So the second plate has information related to dense tissues and bone. So the two plates capture two different views from the same X-ray beam, one exposure. The patient is not subjected to two or multiple exposures. There is no need to worry about motion artifacts. Weighted subtraction of the two images provides various results. I asked for some details and he said he couldn't give me that because it was licensed to a company. And I couldn't get the separate images. So this is the full spectrum, what is called CR, computed radiography. That's a confusing name. What it means, it's a charge plate like in your photocopier, and that is exposed to X-rays. There is a charge distribution that is scanned, and you get a picture. It's not computed, it is scanned. Whereas direct digital means it's a solid state digital detector directly without converting the X-rays to light or to some charge or some other form of energy. So CR and DR are the terms used. Computed radiography, when you have a charge plate and that is scanned later to get the latent image out. Digital radiography or DR, when you have a direct digital solid state detector directly for X-rays. So Dr. McMahon at the University of Chicago developed this, and this is the full spectrum chest image. So you see all the details, bones and ribs and some lung structure and the heart and diaphragm and all that. So this is the bone detail image, and this is the soft tissue detail image by energy subtraction of the two images obtained simultaneously. I asked for details about the weights and the two images separately so I could try different operations, and he said he couldn't do that. It was licensed to some other company. So from the two images, you get the separation of the hard tissue and soft tissue details. And there is a small dark spot here that shows better here than there. That's the normal, typical image. And here you have two separate images obtained by manipulation of the two images obtained in one shot. So that is subtraction, just A minus B. But you can also do that over time. But for change over time, temporal subtraction, time-lapse subtraction of images or scenes, when something is moving and changing, you take one at this instant, another later, and subtract, and you get what changed in between. It could also be a patient's image from today and next year, or this year and last year. But then that's not so easy. It can't be A minus B. You have to register. I would have changed I was joking with my sister-in-law. I was telling my size was 42 before I came, and one day after I came, I went shopping, my size was 44. I said, I'm eating too much here in India. So we change, especially over a year we change. And you can't simply subtract the two images, you have to register. Hopefully there are some marks, some landmarks. I'll talk about landmarks later on the last day or the last 2 days. You take those landmarks and register the image, and you also have to do some geometric mapping, and then you can subtract. Otherwise, pixel by pixel subtraction doesn't work. So time-lapse subtraction of images is useful in detecting normal or pathological changes that have occurred over that period of time. Radiologists do this. Mammograms taken in a screening program. When a woman goes for this imaging today, the previous images from the last year or the previous screening, typically 2 years ago, and the one before that, 4 years ago, they're all brought up and they compare. They don't do subtraction, they just compare. Visually, they do the subtraction mentally. So normal anatomical structures are suppressed and pathological changes are enhanced. That's what you would want. Registration is crucial in temporal subtraction. Otherwise, you get artifacts. You saw in the digital subtraction angiography, even within a few seconds, the pulsatile movement of the vessels will cause artifacts. Geometric transformation techniques are useful in matching landmark features, but you need landmarks for registration. So you require detection of those landmarks. That itself is a big issue. So those are some other topics to consider. So let's look at transforms for enhancement now. Different types of enhancement and different types of transforms, not the Fourier transform, but grayscale transforms. How do you change the grayscale values in an image? Grayscale thresholding is simple. Convert a grayscale image with a range 0 to 255 to a binary image. So put a threshold and say everything below this is not of interest, everything above that is of interest, but just give it one value. I don't care about the variations in the two ranges. So GMN is 0 if FMN is less than or equal to L1, and 255 if FMN is greater than L1. Some L1 is some threshold. You have to determine the threshold in an optimal way. It's a bi-level or binary image. This is bi-level, this is not 1. If it is 1, you call it binary. So essentially you're saying pixels less than L1 are noise or background that I'm not interested in, and gray levels of objects of interest are greater than L1. You can also say if it is less than L1, I'll make it 0, but if it is greater than L1, I'll keep the original value. So the variations are retained. So here is an example, a CT image of the patient I showed earlier with neuroblastoma, the tumor. Here, B is, okay, this shows the Hounsfield unit range of minus 200 to plus 400. Anything less than minus 200 is black. Anything more than 400 is saturated to 255. This is thresholded at 200 Hounsfield units. So the calcified tissues, the bones, the spine, spinal column, and that the examination bench of the CT scanner, they are visible. So you have these bits of calcification within the tumor. Here, values above the threshold are retained in their original value, about 200. So this shows the difference in density between this and that. The calcifications are not of the same density or Hounsfield units as the ribs. Here, they all appear the same. And in D, the Hounsfield unit range of 0 to 400 has been mapped to the range 0 to 255, whereas here it is from minus 200 to plus 400. From 0 to minus 200, you have fat. Water is 0, fat is minus 10, 20, and air is minus 300, 400, minus 1000 would all be air. So if you're not interested in anything less dense than water with negative Hounsfield units, then you may use this 0 to 400. This is a pediatric case. Above 400, there may not be many pixels. 400 is fairly high density bone. Adults will have high density bone, but not a child. So you may say this is enough. And then the details within are seen a little better. You select that, window that, and then look at the details. This is not bad, this is bright, but this is in a limited range. So these are thresholding, these are just rescaling. This is necessary, this windowing is necessary with CT images because CT images will have a range of plus minus 1000 and you can't display the whole thing. If you do, then you'll be compressing all the details. You will not be able to differentiate between soft tissues of various types. So that is called grayscale windowing, which was already done there. In grayscale windowing, you say, well, my image has such a big range, I can only look at small parts. The human visual system can only discriminate between some 30, 40 gray levels. If you have 0 to 255, you say there are 256 gray levels, but it'll be hard for a human observer to separate all of them. More so when you have a CT image with minus 1000 to plus 1000, 2000 values. Can you, in an image that has a range of 2000, would you be able to separate two things that are differing by 1 or 2 Hounsfield units? Quite likely no. If there is a difference of 10 Hounsfield units, maybe you will see. But if you take a small window of the grayscale and expand it, then you can see the difference. So here you say GMN is 0 if FMN is less than F1, some limit. It could be minus 1000. FMN minus F1 divided by F2 minus F1. That's a scaling factor. The range of interest is defined as F1 to F2. That's the range I want. Below F1, make it 0. Above F2, make it 1 or 255. Multiply this by 255, you get, and make them integers, you get 0 to 255. Otherwise, 0 to 1. Later on, that multiplication factor could be something else. If you are doing computer analysis, you may make it 1000 or whatever you want it to be, but for display 8 bits, 255. So greater than F2, 1. So in details of interest in the range F1 to F2 are stretched. So it could be small, just from 0 to 100 Hounsfield units. Stretch it 0 to 255, so you're doubling the differences in the range. So you can see the abdominal organs which are in that range, 50, 60, 70, 80 Hounsfield units. If you keep 0, water as reference, that will be good. So this is linear. So GMN is the windowed image with the grayscale normalized to 0 to 1. F1 to F2 is a range selected for display. Pixels below F1 will be eliminated, made black. Above F2 will be saturated and shown as white. Details within the range F1 to F2 will be displayed with increased contrast, but it depends. What is that range? If your display range is 0 to 255, if you choose minus 500 to plus 500, a range of 1000, then you are compressing. If you choose a small range like 0 to 50 or 0 to 80, and then expand to 0 to 255, you're getting better contrast. So it's up to you as to what you want to do. If you want to see fracture, then you choose maybe 200 to 1000 and let all the soft tissue go away and take only the bone and take 200 to 1000 and map to 0 to 255. You can see fractures better. So this is just linear stretching. So this is the original image. 0 to 0.6 has been taken, mapped to 0 to 1. And 0.2 to 0.7 mapped to 0 to 1. So if I put a threshold of 0.2, some of the low density details have been removed. If I'm going only to 0.6, so some of the high density details are saturated here, but they are retained there. So it depends on what you want to see. The range is, I'm using normalized range, 0 to 1. This is the histogram of the original image going 0 to 255. So you can take some part that you are of interest. So the transformation, one of them is that, and the other is this, linear. One saturates there, the other saturates here. And there is clipping here. The two transformations I described now. Graphical representation. This is the myocyte image. This is what I got originally. And I stretched that 0.2 to 0.6. to 0 to 1. So I can see the details better. There is some improvement of the visibility. Just because this is a very dark image, it's been brightened. And some parts have been saturated here, white. There is no contrast enhancement. It's only stretching. That gives you increased visible contrast, but that is achieved only by stretching the values and selecting a small range to display.